I am not Ken Loparo. This is supposed to be Ken Loparo's spot, but he cannot make it today, so we exchanged the spot. So instead of, he will be doing that on the February 5th. So my name is Vira Chankong. I'm a faculty professor in the system and control programs. So the kind of research that we do let me do it again. Can be described or can be classified very succinctly as the computational engineering in medicine. In terms of the collaborator, oh, sorry. This is a high-tech slave. Okay. We're working mostly with the people in the so-called radiation oncology, both at University Hospital, Dr. Jason Son at University Hospital here. But my main collaborators from Duke, Dr. Jackie Wu used to be here at University Hospital. She moved to Duke Medical Center a few years ago, but we're still doing collaborative work. In fact, a lot of many of my my students have gone there and spent the time working in the lab in the machine there. In fact, one of the students is still continuing as a postdoc, and they are still looking for good students to go and work in the same project over there. All right. The goal of the work, of course, is to support, to help, to use the technology, the 21st century technology in computer and in information and internet to help improve healthcare delivery and management. To help, in other words, this kind of work again is, we're not going to do the work of the clinicians or healthcare provider, but we support them. You know, if you remember the commercial by BAF, right? They said they show this product like cars like this. They said they're not building the car, but they are building. They're making the car better. Okay, they make products that make the car better. So that's exactly what our role is, to support the role of the healthcare provider, clinician, to do the job better. And particularly in terms of healthcare delivery and management. If you think about it, there are two types of activities that the people, the healthcare provider and clinician do. They do diagnosis and they do treatment, therapeutic. Those are the two things. But as you probably know, even with the 21st century medicine and with all the advances in knowledge in physiology, anatomy, and biology, diagnosis and therapeutic practice are still not a perfect science. And they are obviously looking for help, any kind of help that would help them improve the delivery, the healthcare delivery and management. So that is the general area, but of course this is still too broad, so we have to be more specific in what we focus on. So we are focusing on the diagnostics and treatment of cancer. It's not a very pleasant topic, but it is obviously a relevant one. Particularly, we, as our role, of course, to provide this computational technology, information technology to help. These are the kind of things that we normally do. You know, as you can see, that the process, for example, in treating cancer, involve all kind of imaging, both for diagnosis and for therapeutic. And sometimes you want to do that efficiently, and then, there is a new things that Professor G.Q. Zhang is working on, and I'm joining his team to do work in that too, the compressive sensing. And I will let him talk about that on February 5th. Then you have to do image registration for the purpose of adjusting the treatment plan based on the change in the deformation of organs and things like that. Monte Carlo simulation for dose estimation. When you deliver the dose by a machine, usually you have to estimate the delivery of the dose, the strength or the intensity of the dose at a particular spot. And very often you do that by simulation. And then modeling and optimization, particularly for the treatment planning and delivery. When Dr. Jackie Wu was here, we started collaborating using with the gamma knife radiosurgery. This is for brain tumor. In other words, doing the surgery using gamma knife machine. Well, you will see what that means a little bit later on. And then we continue to work, continue our collaboration, working on the brachytherapy of prostate cancer. This is another form of delivery of the radiation. It's not by radiation beams or anything. It's by implanting radiation seeds in the tumor area. Then the work that we've done in the last few years is the intensity modulated radiation therapy, image guided radiation therapy, or arc radiation therapy. Obviously, the last one is the one that is currently is very hot, everybody is now jumping into this. Gamma knife, it's very quickly, in view of time, very quickly, gamma knife is a kind of surgery that use I'm going to use this screen, right? He used gamma knife to pass through this helmet that has about 201 cobalt ray hosts. So everybody is focusing on the same spot. So that's why the area of effective dose is very small. The size depends on the size of the host. And usually there are 4 sizes that are used in practice: 4 millimeter, 8 millimeters, 14 and 16, and 18. I'm sorry. So you have already 4 sizes, and then if the tumor is small, then all you have to do is just focusing on that spot. But the problem is that tumor is usually large, even in the brain. You can have a tumor of about 5 centimeters and so on. So, you have to have several shots Okay, so you do the planning. The task of the planning is to determine, for example, how many shots you need, what will be the location of each shot, what will be the size of each shot, and how long you leave each shot on, because the intensity of the dose depends on how long you leave it on. So the problem becomes an optimization problem that is very similar to what we call a ball packing problem. You pack this ball into this area, so that in the end you would like the shape of the contour of the 50% contour. You have to have at least 50% of the prescribed dose in order to kill cancer cell. So you have to have at least 50% in order to form the contour. But at the same time, you know that if the radiation can kill cancer cell, it also can kill normal cell. So you want to deliver the prescribed dose to the target area while minimizing the damage to the normal tissue surrounding it, particularly if you have a lot of normal tissue surrounding that. So that is become a multiple objective problem. These are the kind of, when you do in 3 dimensions, usually you will have this, what you call a slice of the 2-dimensional images to work with. And so you can evaluate your plan by just looking at this kind of things. So these are, the kind of things that you do in this kind of a planner, Gamma Knife planner. This one we have already done. There's no more we are doing. So let's go on to the next. So this is the kind of optimization problem that you do. You have to choose the number of shots, the size of each shot, the location of each shot, and the timing of each shot. So you can see the decision variables become a mixed integer and also continuous decision problem. So this is and usually, again, you want to minimize the toxicity to the normal cell, to the normal tissue, while delivering at least 50% to the target area. And obviously, you don't want it to be more than 100% of the prescribed dose because it will be overkill. You will burn too much burning. Obviously, the treatment time is very important. You want to minimize that, at least not excessive, and you have to choose it from collimator size. So that's the mixed integer programming problem. The next one, of course, is the intensity moderated radiation therapy. This is the current technology being used now. This is what, particularly for treating some other kind of a cancer, like head and neck or prostate. Okay, so these are the kind, you can see it's a different kind of machine. You have a patient laying here, gantry rotating around, okay, and delivering the dose. This is a kind of head and neck kind of cancer that you have to work with, the kind of images that you have to work with, but particularly this kind of images, you can see that this is a cross section that go. You have a brain and brain stem and so on. Those the red area is the target area. That's a critical target volume. With the yellow, that's a planned target volume. So you do a little bit. You do planning to deliver a little bit more than just slightly larger than the critical target volume. But at the same time, you have a lot of critical organs surrounding it. So you have to be very careful there. The brain stem, the parotid, the mandible, and so on. These are, this is very sensitive. So it's a very difficult kind of planning to do. Optic nerves. Okay, this is a prostate. Okay, so again, prostate is a little bit easier, and then by the way, the majority of cancer for men, of course, is a prostate. So this is it's easier, but it's still difficult because you have this rectum and femoral and bladder that is right next to it, and bladder is very difficult. You can't put any radiation to your bladder too much because that you're going to be very, very uncomfortable if you do that. So it's again very important there. All right, so this is the kind of activity that you do. By the way, there is something missing on the top there. So you start by getting the image, and then you then do you get, let's say, the MRI images. And then from that MRI image, the doctor then prescribe, he or she will draw the contour around the target area that they want. And then you do some kind of image deformation, registration deformation in order to reconcile the image. And then you do the planning, the optimization planning. And then you deliver. The delivery is done by you can see that what we call a multi-leaf collimator system. So and then usually the treatment is done in more than one session, 25 sessions. That means you do it every day. So patient come in for each session, and the problem is that you have a lot of movement. Even though you try to set up the setup accurately, but there will still be error due to patient setup, and also there will be error due to patient movement like breathing, all this stuff that you have to account for every day. So you have to do this. You have to take image every day, and the current technology allow you to do that. So you take the image of the day, and then do image registration to register the image with the with the original image that you do the planning, you use to do the planning. So and you do the adjustment of the plan. So all of these things is done. And it used to be before that what they do, they do what we call a forward planning, meaning that they just do try and error. They just, okay, try this beam, try that this at this angle, that beam at that angle, and see what the contour is. If it's not satisfied, then you go and adjust the beam. So you do that plan. But nowadays, you do what we call the so-called the inverse planning, meaning that you already have the prescribed dose on hand, and you try to determine what kind of delivery, what kind of intensity of delivery of the beam that you should deliver in order to get what the prescribed dose. So this is what the IMRT is all about. In the delivery phase, you use this multi-leaf. Okay, let me go to this section. So the IMRTs usually go like that. You determine a number of angles that you want to shoot the beam, and each angle you have to determine the shape of the beam using this collimator to make the adjustment. So in the delivery process, you have to do this movement of the multi-collimator of the beam. So and do again, that's a multi-objective mixed integer linear program as well. So this is a kind of a collimator, and that 7 beams does a better job, 3 beam not better. Now here is the one that I said is hot. This is what we are working on right now. Here, instead of determining the number of beams, 5, 7, as in MRT, you rotate this thing in a continuous fashion, and you do the delivery and the planning at the same time. This way, you save time, and you also save, you minimize the damage, because usually when you do it the other way, sometimes the treatment is worse than the disease because of the side effect. So here is a way to reduce that. Try to reduce the time. So here, again, all right, I have a movie there. I'm going to just go to my last slide. So here, what do we have to do? Again, go back to this diagram. The first thing, of course, is to do the imaging, both initially and also the image of the day. And obviously, any kind of technology that will do that better is good. Then you do the deformable image registration. So this is another kind of a computational technique that we need to do well. Then you do the Monte Carlo simulation to do the dose calculation there. Then you do large-scale multi-objective optimization. And you have to help decision maker. You present a lot of plans to the physician or clinician, and he or she has to choose it, making trade-offs, and you may have to help them do that. But this is the one that is for the ART system. This is the one that we are using. The task that we have to do here is the modeling part. Again, there are a lot of techniques being now depending on the kind of model you use. And I say that we can do a lot better with that modeling. And then after that, we can do a large-scale mixed integer dynamic combinatorial optimization. You can see this has all kind of this nasty characteristics that make the problem difficult to solve. But this is a very challenging one. Okay, thank you. Thank you very much.
If there's any questions, it'll probably take me a minute to get set up.
Okay, any question? Sorry, I have to rush through. I know that it's already 5 minutes late. Anybody interested in in this, both the application and also in the this theoretical and challenging development of how to solve this mixed integer linear, large-scale mixed integer combinatorial optimization problem very well. And obviously time is also very critical, and as well as the accuracy. So both the time and accuracy is important. How to do that? Thank you.
Okay, my name is Garverick. I'm professor in the EE program here at Case. I'm going to give you a quick overview of the work that we're doing in my research group, which starts from the background of mixed signal integrated circuits. That's where I, oh, thanks. Okay, can you hear me okay? That's the background I started with when I arrived at Case, but I quickly learned that there's one last frontier for analog circuit designers, and that's basically acquiring and using data. So we quickly settled in on a program where we're looking at sensors and actuators and the interface between the real world and the digital world. There are two main thrusts in my group right now. The first is in high temperature sensing, and the motivation is energy and environment. Just about any system that consumes or produces energy gets hot. And if you can put sensors at the point of energy consumption, you can monitor the operation, you can control the operation, you can improve fuel efficiency, reduce emissions, and you can collect more energy in the case of, for example, geothermal exploration. It gets hot when you put sensors down on the end of a drill bit. At present, our primary collaborators in these efforts are with Dr. Marigani's MINO group, who does a lot of the MEMS fabrication and electronics fabrication, I'll show you. And then that particular project, we also have a collaboration with University of California, Berkeley, with Professor Pisano, a MEMS expert. The other main thrust in my group is in biomedical sensing. Obvious motivations, the life sciences and human health. We have collaborators here at Case Western Reserve University, Professor Cheel in biology, studies aplysia, which is a common sea slug. He's basically mapping out their neural system, learning how neurons interchange information to control animal behavior. And that includes Professor Heidi Martin in chemical engineering and Dr. Tabib Azar, who just left our university for Utah. But we continue the collaboration. I also collaborate with Dr. Fleischman at the Cleveland Clinic, who looks at things like intravascular ultrasonic imaging. In other words, putting an imaging device inside a blood vessel. It obviously has to be small, and that's where we come in. And we have some great collaborations ongoing with the VA hospital, which I'll speak briefly about. Believe it or not, there are some common issues here. This is not just a random collection of stuff. All require very low noise sensing. The signals that are produced, whether we're talking neurons or MEMS capacitive sensors for engine control, are very weak. And you need to have electronics right at the point of sense so you can obtain a high quality signal. The point of sense is usually a very, I hate the word, well, I don't hate the word, but I'm a little tired of the word harsh environment. It's been overused, but there's nothing much more harsh than being inside a fuel injector at 600 degrees centigrade or being inside the human body. Those are pretty harsh environments, and there's not a lot of electrical power available, so these circuits have to run with very low energy. And finally, they all involve short-range, point-to-point, wireless or non-contact communication. We're not. I try to avoid the word wireless because we're not talking cell phones and things like that. We're talking about getting a sensor signal from here to here, through the human body or through an engine compartment. Or I'm going to show you one last example where the non-contact distance is actually about a nanometer. We'd love to get more into network sensing, but we're not doing that yet, and that may be a new thrust in coming years. Okay, I've got slides from every student and researcher in my group, and there's a lot of detail which I can't possibly go through. I'm going to put these slides up, as I always do, on the bulletin board outside my office, so if you're interested in details on any of these particular projects, you can take a look there or talk to us. So I'm just going to give you a quick overview here. Anand is joined our group and picked up a project that we have involving sensing of flow, pressure, and so forth inside a fuel injector. in order to obtain the sensor information that you would need to optimize the operation of a turbine engine, for example. In this case, we've got a passive sensor, basically a variable capacitor, on the inside of the fuel injector. The wall of the fuel injector, which is made out of ceramic, is about 1 millimeter thick. And on the outside of that wall, we have some electronics that can pick up the variable capacitance from inside the fuel injector. That's done through coupled induction. And what we do is sense the change in resonant frequency. And so Anand has picked up a project that Amita Patil started, in which we have an oscillator which oscillates at the resonant frequency of this interface. It goes through a simple counter and so forth and on out to a computer so that we can analyze the change of pressure and so forth that's going on inside the injector. Amita Patil, who I just mentioned, is soon to finish her PhD on the subject of silicon carbide electronic circuits. Now, silicon carbide is a semiconductor that can operate at extremely high temperatures. It has a band gap voltage of around 3 volts, so it doesn't turn into intrinsic material until very high temperatures. She has recently tested, I believe, the first monolithic amplifier to operate at 600C. It's a transimpedance amplifier, which means it senses the flow of current from one of these sensor types we've been talking about. She's also got an array of differential amplifiers that would be used with piezoresistive Wheatstone bridge type sensors that they're developing at NASA and also in Dr. Marigani's group. David Tian will be picking up that project, and we want to move it up more to the system level and combine our amplifiers with some MEMS devices that could act as sensors or resonant devices for communications in and out of basically a little wireless package that you could throw somewhere in a chamber that's at 600C, and then we wirelessly collect the information. Now switching over to the life sciences and the health side of things, Cenk Bobby Liu is picking up a project that we began several years ago in which we are developing a wireless sensor that can be implanted inside a plizia, the sea slug, attached to neurons, and then supply data of the neural activity inside the sea slug. We now have a 4-channel chip, which we're trying to get into the animal. But in the past, we've worked with 2-channel chips in which we could sense and stimulate from a pair of neurons in order to help, as I said, map out the activity and the interconnection between the neurons in the animal. This is just a demonstration of an integrated circuit that we developed and some data that we collected. It's pretty noisy looking, but believe it or not, there's some information in there. Those bigger signals are the spikes, the sea slug thinking about something, and the rest of it's noise. In fact, the fact that there's so much noise in there leads us to conclude that we could save a lot of power if we could identify what's important and what's not before we transmit it, right? If we transmit fewer bits, it takes less power. And that's the subject of Ming's thesis, Almost Complete, in which he's developed a very simple threshold-based detection and compression algorithm so that we can take, I know this is hard to see, on the top trace, that's some recorded neural data, which consists of some spikes and some noise. And using a threshold detection algorithm, Ming has been able to detect what appear to be real spikes and then send that out in an encoded, highly compressed format. And then when you reconstruct and display it, there's not a whole lot of difference between the top trace and the bottom trace other than the fact that we've not transmitted all that noise. He's implemented this in a PIC microprocessor. The algorithm's quite simple. And he's also written a Verilog version that generated this VLSI layout. It's about 1 millimeter by 1 millimeter. Mike Likas has recently joined our group and is helping us to improve our antenna design. Better antenna means that we can pick up a weaker signal, means we can transmit with lower power. And Michael's been looking at things like MRI systems, believe it or not, have very sophisticated, well, I guess it's not surprising, but MRI systems have very sophisticated phased array antennas so that you can maximize the coverage over, say, a volume of an aquarium that we use in our experiments. And Michael's looking at techniques like that to help us improve our antenna design. Yingying Wang has joined our group, and she's helping us look at the other part of the problem. How do we get power into the sensor? Well, at present, in the Aplysia case, we're using a camera battery. The Aplysia is a pretty big animal with lots of empty space. In the future, we want to use a much smaller rechargeable lithium-ion battery, and Yingying is helping us develop a high-power Class E amplifier that will allow us to radiate enough RF energy into the aquarium that we could recharge the battery during times of rest between experiments. And finally, in this category, Jeff Barberitz is taking some of our 2-channel chips, combining them to make a 4-channel system so that Dr. Cheel can look at 4 neurons at a time, stimulating, recording, looking at the interaction between the neurons. In the subject of human health, a different project, Dan Howe is building an electronic bandage. Basically, an electrical stimulator made on a flexible PCB that could be adhered to the surface of the skin where you might have a wound. They call it a ischemic wound. They're basically starved of oxygen, and by applying electrical stimulation, you can speed the healing process. It's not well understood, but Dan's device will help us do more experiments and understand the healing process a little better. That's a picture of a rabbit ear, which was a subject of this experiment. In the future, we're going to be looking more towards working with rodents, and he's developing a smaller version. Steve Majerus is working on a VA project in which we're developing a wireless device that can be inserted into the wall of the bladder and measure the bladder pressure, which could ultimately be used to help an incontinent sufferer determine when they need to go do something. And in the meanwhile, it'll greatly facilitate the experiments that they do at the VA to help understand incontinence. So, it's unfortunate this picture doesn't show up a little better. It's pretty neat. This is Steve's circuit board. This integrated circuit die, which is about 2 millimeters on a side, is underneath that black dot of epoxy. There's a pressure sensor, a Wheatstone bridge type device underneath that black dot of epoxy, and they're connected together on this little PCB, which is almost exactly the size of the Lincoln Memorial. That's a penny, of course, so it's pretty small. Okay, last thing, Dr. Liango Shen joined us this year, and he's working on that 1 nanometer non-contact sensing project I'm talking about. This is a project with Dr. Tabib Azar in which we are trying to fabricate dots and lines on the nanometer scale using a system that's closely related to atomic force microscopy. And our goal here is to develop the electronics that will allow a MEMS cantilever beam to maintain a spacing of 1 to 10 nanometers as it's moved across the surface. And of course, there's surface roughness, and the electronics has to adjust the height of the beam as we drag it across, and that involves sensing that 1 nanometer spacing and then holding it fixed as things move. Okay, I need to stop here and let our next speaker start. Thank you. Again, these will be posted outside my office if you'd like to see more. Memory stick? Okay, just plug it in one of those holes. Oh, yes, thank you. That's the microphone.
Okay.
And you better get that too.
Okay.
Oh, right. Thanks for that would have been embarrassing. Okay.
Oh, that would be okay.
Let me see, it didn't pop up. Oh, must be this.
This one. This.
This one?
Yeah. Okay. I'm Meral Ozsoyoglu. I'm a professor in the CS program, and I'm also the department chair currently. G, okay. Yeah. First of all, I want to talk about my research identity. That is, my research interests are in database systems. And in terms of basic research, I'm interested in data models, query languages, especially for graph structured and hierarchical structured data, and also query optimization and index structures, specifically for similarity search, not exact match search. And what we do in my group is basically we apply these basic techniques and basic research contributions to several applications. Like one of the applications is Pathways Database System. That is the, I will talk about that project in a little more detail. And also like similarity search for genomic sequences, and XML, RDF, query processing and optimization, and also pedigree data management for medical informatics. And basically in here I say optimization, but query optimization means efficiently processing queries. It's not really optimization, true optimization. In terms of teaching, I was teaching data structures and files and database systems courses and algorithms courses, but I'm not teaching any courses this year. Hopefully next year I will start teaching data structures and files. Now I want to talk about the research projects. So this is the summary. I don't know how much time we'll have, so that's why I put everything in one slide in case we won't have time to go over the rest of it. First project is the Pathways project. That is the Case Pathways database system. This is a collaborative work with Professor Tekin Ozsoyoglu and also Joe Nadeau from medical school. Basically, we modeled Pathways as a graph structured database and work on querying and visualizing pathways in multiple abstraction levels. And now this is an NSF supported project, and the next phase of this is using Pathways, PathCase, as a systems biology tool that is not only storing the wirings of metabolic pathways, that is what are the components, what are the molecules, what are the enzymes or genes. We are also modeling or storing the models, dynamic models of pathways, how these pathways work, and stoichiometry of the components. And also using pathways to visualize and query these and also simulate with the more advanced goal of using this as a tool for systems biologists so that they can come up with models for pathways more easily, more effectively, and more efficiently. So that is the ultimate goal. And in terms of similarity search and distance space indexing, work on proximity search for genomic sequences, and also work on optimum nearest neighbor search and generalizing distance space indexing for almost metrics. In fact, this is also applicable. This is applicable to sequences as well as genomic sequences, but also for images and image searches. For multidimensional search, typical search methods do not work well, so you end up using specialized index structures. I'm really interested in query processing, query optimization. Actually, that was my PhD thesis as well many, many years ago, and I still continue on that. And we work on XML query processing and query processing with materialized views. As the size of the data becomes large, efficient query processing becomes more and more important because people want the results right away. And a new challenge in this is RDF, that is Record Description Framework. In fact, typically in relational databases, those of you who have taken database courses will relate to that, there is a schema of the database. And so when you are doing the query processing, you heavily utilize the schema information. so that you can optimize. But in case of RDF, there is no schema. Everything is just a triple. So object, subject, and predicate. So the good thing about this is you really don't have to worry about the schema changes and restructure the data or anything like that. But the disadvantage is you really don't have the data semantics built in the schema. So you have a very large collection of triples. So then processing these queries on RDF becomes more of a challenge. So we are working on coming up with efficient, scalable ways of processing RDF queries. Actually, I have just a PhD student graduated. His thesis was on this, and he's now in Microsoft. But there's still a lot more work to be done. The next project is the pedigree data management. That is about the genealogical data. And now with the more recent interest on medical informatics and medical records, it is very important to be able to link this genealogical data to the medical records. And pedigree data has special characteristics. It's hierarchically organized. It's not like tabular data like medical records. So the idea is to be able to query this and also come up with scalable querying and also analysis. That is, for example, there's an inbreeding kinship coefficients, identity coefficients that medical researchers use for genetic analysis. And these are typically computed by recursive methods. And as the size of the data becomes large, these become not scalable. So we recently come up with a method using path computation and some sort of an encoding. If we have time, I will be able to go a little bit over that more. So that really significantly improved the efficiency. So the next step is to tie the pedigree data, medical records, as well as genetic data and gene sequences. Okay, so basically for the synergistic opportunities for systems biology, we are using PathCase as a tool for systems biology. For data mining, similarity search, distance-based indexing applies very well. And information retrieval, we work on semantic query evaluation and context-based retrieval. And for medical informatics, as I mentioned, scalable querying, medical records, and data integration, integrating medical records, genealogical data, as well as genetic data. In general, I am working on research projects involving management of data, modeling, querying, efficient and scalable retrieval of large heterogeneous collections of structured and unstructured data. Actually, large is very important here because you can't do anything if your data is small. The challenge for us, for the techniques we have, is to be able to do that in a scalable way for very large collections of data. Okay, so for the I think I have a couple more minutes. For the PathCase project, this is a web-based exploratory querying and visualization tool for biological pathways, as I said. If you go to this link, there's all kinds of documentation and tutorials, as well as information about this. And the collaborators, as I say, Cenk and Jornado for the earlier version of this. And now we are collaborating also with Marco Cabrera from medical school for the systems biology version of PathCase. And there has been several students and postdocs work on this project. Some of them are still working on it. So I list some of those there. And so in case of PathCase, as I said, we are working on metabolic pathways. So this is a part of a pathway. There are processes, there are metabolites and enzymes. So we represent this in the form of a relational database. Conceptually, it is a hypergraph. There are hyper edges. Edges are the processes, and the vertices are the metabolites. But in the lowest level, it's a relational database. So querying and visualization is based on the graph conceptual view. So basically, for the data sources, see, we are computer scientists. and engineers at most, but we cannot generate the data. So we have a academic contract with Cenk from Japan, so we download their pathways. We also have another version of this for biopsych human pathways, and also we have some featuring sample pathways. Those are just we downloaded from, we just stored ourselves from a metabolic pathways textbook. So there are lots of data, and basically architecture is a typical client-server architecture. And so this is a first page of path case. So you can use this for browsing pathways, organisms, genes, proteins. You can also have built-in queries or ask queries using advanced query interface. So there are many different versions of querying, and of course we are using the database techniques for having efficient queries. One interesting query is the neighborhood query in a pathway. That is, you can click on a particular process or metabolite and query the neighborhood of that in terms of whether the processes and metabolites within 1 step, 2 steps, or whatever. So this shows the example of it. So you can indicate how many steps you want. Basically, you can't remember the names of these things, so we have a drop-down menu that you can choose. And the results are shown as a tabular output as well as a graphical output. So this is the source process, and it shows the first step metabolites, and green is the second step, and then blue is the third step. And you can also do this in multiple levels of abstraction. That is, you can visualize, since you store this everything in the database now, you can visualize each pathway as a node. And then we explore the connected pathways. And then you can collapse the pathways into a single node or expand it, and so on and so forth. So let me talk very briefly about the pedigree data. This is focusing on modeling pedigree data and coming up with query languages for querying pedigree data, and also doing the query processing in an efficient and scalable way, as well as computing inbreeding coefficient, kinship, and identity coefficients. And we collaborate with Robert Elston from epidemiology and biostatistics. Students involved in this project are Brandon and Cenk, Fatih, and Stephen Mays. And first of all, pedigree is a tree that shows the family's genealogy. And using pedigrees, you can see how a particular trait or disease has been inherited. It is very useful in analyzing hereditary diseases. And the problem is that there are very, very large pedigrees, and these pedigrees are becoming larger as well, because collecting the data becomes cheaper and also easier. But then the challenge for us is to be able to effectively utilize this data. And currently this data all stored in flat files, and there are only simple ID indexes, and there's keyword searches, ad hoc SQL, predefined relationship searches. The problem is data is very large, and there's only limited querying, and it is not scalable. So for example, simple sample pedigree queries find all ancestors of an individual, find all first degree, second degree relatives of a proband individual who were diagnosed with cancer before age 50. Or even there are more complicated ones as well. So what we did is we just modeled this. Actually, we collaborated with Dr. Church from Cleveland Clinic on this as a directed acyclic graph again. And we used I'm sorry. And okay, we define a language called PQL that makes these queries very easily. For example, for individuals, ancestors is represented in this. It is really similar to XML expat query language. And then we store the data in relational database, apply some sort of an encoding to speed up structural steps, and then convert it to SQL and evaluate. And this also gives us, okay, this is the coding of the diagram. And the idea here is that when you do this coding, you really don't need to get the data. You can look at the individual node and the codes and find all the ancestors and get only those ancestors. So that is how we have the speed up. For the identity and kinship coefficients, this is also very important. It shows how individuals are related with each other, how the genes are passed. And in fact, there is a recursive formula to calculate this. So what we come up with is that we come up with a pad counting formula and use node codes that we originally developed for multimedia presentations actually many years ago, about 10 years ago. And then using node codes on the pedigree and pad counting technique, we came up with a very scalable method for evaluating kinship and identity coefficients. So, and we also showed the effectiveness of our system with experiments on real data. And we are still working on this project. And basically, that's all. Thank you.